Hello everyone, in this video I will discuss about humoral immunity. So what is humoral immunity? Humoral immunity is basically an adaptive immunity. So this is humoral immunity. Humoral immunity is basically a part of adaptive immunity. And in this humoral immunity, mainly B cell involved. So B cell mainly involved in this humoral immunity. So what basically B cell do? B cell mainly produce antibody, and this antibody then inhibit the active site or inhibit the pathogen particle to bind with the cell. So this is basically the summary of humoral immunity. So if I again say that humoral immunity is an adaptive immunity, and here mainly antibody produced, and this antibody is produced by B cell. Okay, now let's go to the next page. Okay, so B cell development and maturation. So as I said earlier that in the humoral immunity mainly B cell involved in this process. So for the activation of B cell or for proper function, B cell need to be matured and developed. So now let's look at this, that B cell mainly originate from the hematopoietic stem cell, and this hematopoietic stem cell is mainly located in the bone marrow. So this is basically this whole thing is basically bone marrow, and in the bone marrow there is many stem cells which can convert into many other types of stem cells. Okay, so B cell originate from hematopoietic stem cell, and then it have to be get mature, because when it get mature, when this B cell is get mature, suppose those are B cells in the bone marrow, and when this B cells in the bone marrow get mature, it contain this receptor, and this receptor is called B cell receptor, or shortly we say that BCR. BCR means B cell receptor. So remember that B cell originate from hematopoietic stem cell, or it develop in the bone marrow, and it also get mature in the bone marrow. But T cell, suppose for the T cell produced in the bone marrow, and it get mature in the thymus gland. We know that, we learned that in the cell mediated immunity. So here B cell mainly produce in the bone marrow, and then it get mature. So why this maturation need? This maturation is need for the receptor, and this receptor is called B cell receptor, and this whole cell is mainly B cell, or now we can say that this whole cell is mainly mature B cell. Mature B cell. Okay, so when the during the maturation of B cell, there is mainly two selection happens. First one is positive selection, and the second one is negative selection. So in the positive selection, this mainly uh, we this production of B cell receptor is mainly positive selection. Positive selection. So this positive selection mainly ensure that B cell contain the B cell receptor, and this B cell, suppose this is, assume that this is B cell receptor, and in this B cell receptor, antigen mainly bind, or it or antigen, or suppose any pathogen get into our body, and this pathogen mainly attached with this B cell receptor. So obviously for the functioning of the B cell should contain B cell receptor. So for this reason, this selection is called this selection is called positive selection. And for the negative selection means that those B cells, suppose our body also has antigens. So in the negative selection, our body cancels, or our body destroys or eliminates those B cells which is suppose which is active for our antigen, for a body antigen, or for our self antigen. So here let's look at this line, that in this negative selection, our body basically eliminates self-reactive B cells to prevent autoimmunity. So if the negative selection doesn't happen, then our own cells, or our own, suppose, protein will get destroyed by this B cell. So in the negative selection, our body mainly kills or destroys the B cell. Which types of B cell? Those B cells that are reactive to our body's own antigen, or own good antigen, or own functioning antigen, which is mainly need for our body. Okay, so now mainly this humoral immunity, or mainly B cell, for the B cell maturation, we obviously positive selection mainly should occur. And when the B cell get mature, it contains B cell receptor. Then it went to the lymphoid organs, and why it went to the lymphoid organs? Because it goes to the lymphoid organs for the searching of different types of antigen, mainly foreign antigen. It mainly search foreign antigen in the lymphoid organs, which who search this antigen in the lymphoid organ? Mature B cell. Okay. Now let's see the B cell activation mechanism. This is the main part of humoral immunity. Uh, so now let's see this, that this B cell activation mainly, B cell mainly in humoral immunity, B cell mainly activated by two ways. One is T cell dependent activation and the other is T cell independent activation. So mainly we learn about, or mainly the B cell, or mainly humoral immunity means T cell dependent activation, because this is the most important activation for the humoral immunity. Okay. Now let's see this. Suppose in the T cell dependent activation, first B cell, B cell receptor, BCR, is get attached with the antigen. This antigen mainly, this is foreign antigen, foreign antigen, just like suppose virus or bacteria. Okay. Okay. B cell is called antigen presenting cell because B cell can present the antigen. Antigen means that foreign antigen that B cell can engulf. Mainly engulf, B cell can take the virus particle into its own cell, into the cell, and then it fragmentize this virus particle. Then it can show these fragments to its surface by MHC class 2 molecule. So for this reason, B cell is also a antigen presenting cell. Okay. Now in the humoral immunity, or in the T cell dependent activation of the humoral activity, so first what happened is this B cell receptor is bind with the antigen, and antigen means this is foreign antigen like virus or bacteria. After the binding, or after the correct binding, this virus or this bacteria or this pathogen can enter into this B cell. Okay. Now then when this antigen enter into this B cell can fragmentize the virus particle. Suppose this is virus particle, and when this virus particle get into the B cell, then the B cell can fragmentize this virus particle. Now this small fragment can now this small fragment is formed into a vesicle mainly, and this vesicle, in the vesicle, the MSC class 2 molecule, here MSC class 2 molecule bind with this fragmentized antigen, and this antigen basically mainly come from the virus or bacteria. Okay. After the binding, the MSC class 2 molecule with its antigen, or we can say that antigen, we can say this, that MSC class 2 antigen complex, and this complex then come into the surface of the B cell. So here what mainly happen is this MSC class 2 molecule and this violet color thing is, this thing is MHC class 2 molecule. This MHC class 2 molecule now showing the antigen particle. Okay. Or this MSC cluster molecule mainly present the antigen particle to other cells for more stimulation or for more immune activation. Now let's see that. So here what is happening, that MSC cluster molecule in the surface of the B cell is presenting the antigen or virus particle or pathogen particle. Okay. When this MSC cluster molecule is presenting the antigen, then helper T cell get activated. In the helper T cell, or in the surface of the helper T cell, there is a receptor called T cell receptor, and this T cell receptor mainly bind with the MSC class 2 molecule, and it get attached with the MSC class 2 molecule for this antigen. So here, in this whole scenario, main thing is helper T cell bind with the MSC cluster molecule, or helper T cell receptor bind with the MSC cluster molecule for the antigen. And there is also another binding, or we can say that this secondary binding. And this secondary binding, there is also a receptor in the B cell called B7, and with this receptor, the T helper cell also has another receptor for against this receptor is CD28. So this binding ensures the stability of this whole binding process. Okay. After this binding, the helper T cell get activated, and when the helper cell get activated, it release cytokines. So these things are cytokines mainly. So after the binding, with which binding, with this MSC cluster molecule antigen complex binding, the helper T cell get activated. After this activation, helper T cell release cytokines, and these cytokines then activate, and these cytokines, suppose then those cytokines then activate the B cell. Okay. So this is a just like a form a cycle type thing. Now let's see this is our B cell, and and this thing is suppose cytokines, and this cytokines is mainly released by the cytokines. Suppose this cytokines is mainly released by T helper cell. The cytokines is mainly released by suppose T helper cell. T helper cell. So this T helper cell release the cytokines, and B cell surface also contain this thing. This thing is cytokine receptor, also contains cytokine receptor. So what happen next, that cytokines will obviously bind with the cytokine receptor in the B cell, and when the cytokine bind with the cytokine receptor in the surface of the B cell, what happen is that this B cell get activated, and when the B cell get activated, it converted into two types of cells. Remember that when cytokine is bind with the B cell, this B cell gets activated, and after this activation, B cell mainly form into two types of cells. One is plasma cell, and the other is memory B cell, and the second cell is memory B cell. So at the beginning, or at the first part of the humoral immunity, we knew that in the humoral immunity mainly antibody is produced, or mainly antibody is involved in the humoral immunity. So this antibody is mainly produced by plasma B cell, or plasma cell, or we can say that plasma cell. So always remember that this cytokine is released by helper T cell, and then the cytokines is bind with the cytokine receptor in the B cell, in the B cell surface. After this B cell get activated, and when the B cell gets activated, it converted into two types of cells. One is activated plasma cell, one is plasma cell, and the other is memory B cell. And this plasma cell is mainly produce different types of antibody. This can be IgM, or this can be IgA, and other types of antigens, and other types of antibody. And this memory cell mainly, remember, this memory cell mainly, this remember the foreign antigen. And this memory B cell is mainly function in the second exposure, suppose second exposure of the same antigen. And this activation will obviously much faster than the previous B cell, because this memory B cell is mainly remember the foreign antigen, the specific foreign antigen, which mainly for that antigen this memory B cell is mainly produced. And after the activation of plasma cell, this plasma cell release antibodies, and antibody mainly, remember that antibody don't directly kill the pathogen, it mainly bind with the pathogen. Suppose after this release of antibody, suppose this is our pathogen, that green color thing is our pathogen, and when this antibody is produced, the antibody here the antibody, suppose let me change the color. Here antibody bind in the surface of the pathogen. And now what will happen, that this pathogen now cannot penetrate or cannot bind with our normal cell. Suppose this is our normal body cell, normal cell, this is our normal cell. And when this antibody bind bind with the pathogen, or mainly this binding is happen in the surface of the pathogen, then this pathogen cannot bind with the receptor of the normal B cell, or sorry, of the normal cell of our normal body cell. And this is the main thing of humoral immunity. And there is another thing, that when antibodies bind with the pathogen, it mainly attract the macrophages. And macrophages mainly, what is the main function of macrophage? It mainly do phagocytosis. Phagocytosis. So suppose this is macrophage, and this macrophage then engulf this complex, this antibody antigen complex, or this antibody pathogen complex. And then after the phagocytosis, the macrophage basically kill the pathogen. And another thing also happened, that after the antibody and pathogen binding, it also creates complement, or it also stimulate the complement system of our body. Okay, this is the whole process of the T cell dependent activation. And now let's see the T cell independent activation. The T cell independent activation is mainly not that serious, or cannot produce that much antibody if we compare the T cell independent activation. So in the T cell independent activation, what happens is that B cell don't need T cell for the activation. In the T cell dependent activation, what we see that B cell need T cell for the activation, because T cell release cytokines, and these cytokines helps in the activation of B cell. But in the T cell independent activation, the B cell don't need T cell for the activation. And this T cell independent activation, obviously B cell will get activated. Obviously B cell will get activated. And here mainly polysaccharides and lipopolysaccharides are involved in the T cell independent activation. And this polysaccharide and lipopolysaccharide obviously comes from the pathogen. It is mainly pathogen polysaccharide or lipopolysaccharide. And in the T cell independent activation, what mainly happens is, suppose this is a B cell, this is B cell, and we know that B cell is also a antigen presenting cell. So what B cell do is, B cell again B cell will engulf a pathogen, then it will show the pathogen particle in its surface. Then here another B cell will get activated. Suppose this is another B cell, here another B cell will get activated by this pathogen. So here mainly B cell helps to activate another B cell. And this is mainly a cross link, or this is a cross link mainly happen in in the T cell independent activation. And this is not cross link, but this is also another thing of the activation of B cell. And in the cross link here mainly, suppose this is a pathogen, and with this pathogen mainly two or three B cell receptor will bind. Suppose this is a B cell, suppose this is a B cell, and this is another B cell. And B cell contain this B cell receptor, and this B cell contain its B cell receptor. So here both B cell receptor bind with the same antigen, and this is mainly called cross link. And for this cross link, here B cell also can get activated. And this is the whole thing of humoral immunity.